One. Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny.
My name is...
In previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week we meet to talk about microbiology, and we're back after like a little bit of a hiatus with our deep dive this week, where we're doing going figure by figure in a paper that we chose during a poll maybe like a month ago.
Oh god, the poll was, I think, this has been delayed a long time because we've been ill. Well, I've been ill a lot, and now Danny's also been ill recently. So we're going to soldier through. We'll talk more about our respective illnesses next week. Now this week is for the deep dive.
Yep. You can follow along with any of the papers that we discuss on either week using our shared Zotero library linked in the doobly-doo below.
Yeah. And if you have any questions, please feel free to dive into the comments and we'll try to respond to you as best we can. And without further ado, boy, do we have a show for you. Yeah, today we're going to be delving into the science of things like mayonnaise. My slides aren't working. Mayonnaise. And also we're going to be looking at the physical opposite of mayonnaise, potentially. This strange substance called oobleck. And also we'll be looking at the significance of what the notion of rotating an Oreo might have on studying rheology and the structures of viscous materials. And finally, the importance of suspension bridges and how they eventually might break. And all this is in favor of trying to discover the most important thing of how to 3D print bacteria. Yes, this is all going to be connected together somehow, and we'll get to that during the show. But without further ado, we're going to be looking at 3D printing with bacteria.
Yeah, this paper was titled Programmable Microbial Ink for 3D Printing of Living Materials Produced from Genetically Engineered Protein Nanofibers.
Yeah.
But before we get to all of that stuff, I think we have to do some, we have to understand like what exactly is 3D printing.
Yeah. And why would we need to oh, good grief, 1 minute. And why would we need to do it? So the big thing, why? Why is 3D printing so cool? Well, so let's say we're talking about like living things, like kind of stem cells, stuff I mean, as a microbiologist, I see the world through a petri dish. Everything is grown on a petri dish, but I've figured out over the years that people aren't like that. That our organs tend to be more three-dimensional.
Yeah.
And so, yeah, you can't necessarily get that from a petri dish.
But like I guess like the cool thing is like, but you can grow all these different types of cells. That's cell culture, right? We've probably talked about it a couple times. But those cell cultures, they aren't organs because of that, or they aren't even tissues, actually, I would say, because they lack that three-dimensional structure. They also lack the complexity, right? Like inside of an organ, it's not just one type of cell. It's not a homogeneous thing. It's a whole bunch of different types of things. And the thought is that 3D printing can help build up that three-dimensional structure. And also, just like how you change out inks for different colors, you could imagine, again, this is all just conceptual, the dream, if you will, the technological dream. You could change out those inks for different types of cells, and you would be able to recreate some of that complexity.
Yeah, indeed. And so I think we've got an image here of some cells that were like placed, I think.
Yeah, so I think that's one of them is vasculature, the top one. So like that idea that, there's tiny capillaries that infiltrate our tissues, and that those things are spreading oxygen. Those structures, you could print them, and this is an example of them printing something like that. So you can create like very thin lines. And then I think let's see if I have it hiding over here. So eye that is also a vascularized, but this one is like a bone, like a type
Right.
of bone. I guess. I mean, I don't really. I'm not a tissue expert, but like bone also has to be vascularized in order to develop, and so like that's an example.
Yeah. And of course, like, so that's really important for like wound healing. So if you've got like a bone that's missing a part, then naturally you'd want to have a 3D printed part that could still get vascularized and behave like real bone. So yeah, printing different cell types is very important to that.
Totally.
So yeah, I mean, so when we talk about 3D printed biofilms, there are lots of different like possibilities
This
out
is
there.
this is the
So
quintessential engineering way of looking at stuff, right? Is to engineers want to parameterize everything, turn things into numbers so that they can run optimization calculations and understand like their design space. So this is like from an engineering from a paper about like 3D printing. This gives us an idea of all the different types of uh, engineered materials that could exist within biologically engineered materials that have like cells, but they could also have abiotic components as well. You don't have to print just living things. Cells also live in extracellular matrix that's made up of different types of proteins and things like that. So that's the artificial scaffold style. And then there's also, and then the other axis here is that cells are growing objects, so that you can have the cells grow into the structure. And so basically your favorite 3D printing, or actually 3D printing fits in this matrix, right? Your favorite way of creating complex 3D structures using biology will fit somewhere in this matrix. And that's where that's, I guess, how engineers split up this whole field to look at stuff.
yeah. So like we are talking before about living cells in artificial materials. So the idea is you have your living cells, but you had it in a polymer hydrogel. And then furthermore, there's a fabrication process. So you kind of print out, so you've got like your kind of your bacteria are floating in whatever medium you've done. So I say some kind of jelly, and you can print out to do some 3D printing. And from there you move to your application scenario. So like you 3D printed a patch that produces antibiotics, like a living plaster, perhaps.
Yeah, absolutely. A poultice,
Yeah.
just to be kind of like fantasy style. But no, people still make poultices. But yeah, like that's I think one of the hopes of this sort of technology. So this is a very specific way. Like 3D printing would be a specific modality to create these types of structures. And so diving deeper into 3D printing itself. Oh, actually, I guess there's a whole bunch of different ways of printing things. But actually, what they'll focus on in this one is extrusion 3D printing.
And that's probably the most easiest one to understand, because essentially, like, it's squeezing stuff out of a nozzle. Like, we all do that when we're working with sauces like ketchup and barbecue sauce, or
toothpaste,
whatever. Yeah, toothpaste as well. Yes, not the greatest of sauces, but it is up there.
Yeah, and so actually, people might be you may be intimately familiar with the engineering parameters that have to go into these types of squeezing into a machine that would do this type of action. But of course, in order to, I guess, talk about those exact engineering constraints that the authors were concerned about, like, we want to explain what that is. And this whole realm is about rheology is one of the ways they talk about it, or viscosity is something you've probably heard thrown around.
Yeah. And so, like, full disclaimer, we are microbiologists. I don't have a PhD in physics, so I'm going to try to explain this kind of relatively simply. So, like, viscosity. So, like, the idea that of running water, of like how honey is more viscous than water because it kind of sticks to the end of things. So, like, kind of a quick demonstration with. So, let's say that my face is a fluid of some sort, and I have a book. There's a boundary layer here. And so what happens if I put my face to a book and move it? The bit of my face that's next to the book will keep moving up, but the rest of it doesn't, because my face is really viscous. So the same applies to liquids, where if I was less viscous, it would stretch a lot more. But at the moment, my face isn't that. But yeah, let's demonstrate it through a diagram. Because my face isn't the best scientific explanation for things. You might know this by now. So yeah, viscosity, like we can imagine like water flowing through a pipe as a series of layers that kind of stick to each other and the edge of the pipe. So as it flows through, these layers will kind of move away from each other. So you'd kind of get the parts of them like kind of moving. So like the more viscous a liquid is, the more it clings to itself. So the less it clings to itself, the less viscous it is. So let's say we have two liquids. One's a bit viscous, one's really viscous.
May I pause for a second? I think yes. this is like this is the way, right? This idea that physicists want to make everything perfect in order to explain like all the pieces that work together. So in this like perfect scenario, that like the liquid could be thought of as like separate layers in of itself, right? Yes. Like, and those layers each have interactions with each other, and so that's like that's not real, right? Like it's not like fluids actually have these layers, but it's helpful to make it discrete in this fashion, because it gives us a way of parameterizing and like putting numbers to those different things. So like we'll act as if the liquids, right, are split up into layers that interact with each other. And then through looking at how a viscous liquid versus a not so viscous liquid acts in this way, we'll have some insight into what we can add numbers to.
Yeah, completely. Because real fluids, they get turbulent. They have all these other things that are really complicated that I can't explain at the moment because I just don't have the knowledge to make. So this is the most simple. But
also, physicists don't want to explain it. Like, I mean, some physicists want to explain it, but like for intensive purposes to understand rheology, like it's that explanation is not required. They don't want to dip into turbulence, even though like that is something that happens in liquids, right? We know that they can swirl and stuff like that.
Yeah, I mean, any physicists like who are watching this, we're very sorry for speaking for you at the moment. But I don't know what your hopes and dreams on. I assume physicists want lots of things. But
oh yeah.
But yeah, for the intensive purposes here, I'm breaking it down as simply as to the level that I can understand it. Anything higher than that is going to be very hard for me to explain. So consider for this example that imagine a fluid is actually a series of layers and not this turbulent. The explanation here isn't going to be complete, but it's going to give you an idea. So let's say we apply a force to these things, something called, they're going to call it a shear stress here, which again, the force causes the shear stress. That's the kind of the amount that a liquid bonds to itself. And something that's really viscous won't like displace as much as something that is not very viscous.
In this example, the shear stress, it's like a pipe, right? So the shear stress is flow in this instance. This is just the liquid flowing through a tube, and right, the liquid that's closest to the sides of the tube has the most friction because it has to interact with that tube, and then it has less as it like interacts only with itself.
Yeah. And like the shear rate is another thing we look at, which is the amount that a liquid is displaced by that shear stress. So
so I actually maybe shear rate is better to say flow, and shear stress here is like pressure, the pressure in the pipe, something like that.
Yeah, that's kind of what because these are like the specific terms that are devised by the material scientists who work on this. And the reason I'm using these terms is so that you can kind of understand what this graph is about, because this is kind of an important graph to understand when we're talking about materials. So you've got Newtonian, where you've got, if you add more force, you're expecting something to stretch more. That makes sense, right?
Yeah. I think that would be water, right?
Yeah, that would be water in this example. But then you've got like things that kind of have a much their shear rate kind of, they become even less viscous the more force you get up on them. So the example here is tomato ketchup. where I don't know if you ever had to shake it out of a bottle, but you need to like really like shake it, and in order to but the more you kind of force it, the more you force you put on it, the more it becomes like a fluid. So in a squeezy bottle, it behaves like a fluid, but then once it's on like kind of your plate, it kind of gets more jelly.
I think that a really nice example of that might be like how when ketchup comes out, sometimes it's like a drop, right? Like you put all that force in, and like a big blob comes out, and like that force had thinned it, so it'll like stretch out, right? You'll get a stretchy kind of shape. But then as soon as it sits down, then it just stays in that shape. It doesn't like, it doesn't splat. Only the fastest moving part is splatting, and the rest of it kind of just stays in the shape that it was, something like that.
Yeah, exactly. So if that was like water, you'd see it spread out on the plate, but on this section, you see it kind of falling in that ball because it's becoming more viscous when the flow gets lower.
Like you were able to increase its viscosity for it to move, but that was only through the force. Once the force is gone, then it holds itself. Yeah.
Then we've got the opposite of ketchup, which is oobleck, where you add more force to it, it'll start to kind of bunch up and stick to. The more force you put on it, the more viscous it gets.
Yeah, and that one's really weird. Like that's a really strange GIF. But like I've also seen people like you can stand on it if you're like always tapping your feet on the surface of it, you'll never sink into that liquid. It has like that's a very strange behavior, but again, one that occurs in this range of different ways like objects can be.
Yeah. And then you get materials that are very hard all the way up to and are solid until you kind of have a break point in stress, and then they are able to flow out. So these things are called Bingham plastics, and the most notorious version of a Bingham plastic is the mayonnaise. Yeah, we've entered the mayonnaise. We need to get out of the mayonnaise zone and talk about more things. But sometimes, mayonnaise. We're not going to be talking about the mayonnaise zone so much. Oh yeah, just for reference, most of these PowerPoint slides we're doing to self off the background are not from the paper. They don't talk about the mayonnaise zone in the paper, unfortunately, or fortunately. I think fortunately.
Yeah, it was our own like trying to understand why it's so important, or like what is the significance, right? Like they're claiming that they have this great ink, but like to understand that significance, you got to dip into Wikipedia.
Yeah. I spent quite a lot of my time being ill learning about viscosity. And why is this important for 3D printing? Well, for 3D printing, we want a material that's able to flow through your jet in order to be printed out. But then once it's been out there, you want it to stay in place.
Yeah.
You want it to be solid.
Yeah. So you want it to spread while it's under shear stress, but you don't want it to spread when it's not under shear stress. Yeah, so you want basically the most ketchup-y ketchup.
Exactly. So the term we're using is pseudoplastic for this. We want something that's a very good pseudoplastic.
Yeah, or the other one people say it's called shear thinning. Shear thinning.
Yes, shear thinning.
Shear thinning. Yeah, but, and so the authors have actually done something very similar to this before in the past. Specifically like this example where they have living cells that make, so E. coli makes biofilm. E. coli makes tons of biofilm things, but one of the compounds that is well known, it's proteinaceous. It's called curli because it looks like, as you can see here, it kind of looks like curls under the electron microscope. And so the authors have worked with this before, and they think, oh, this could be a nice ink medium because the cells make it themselves. We also want to put cells in our ink. So like they're just seeing a lot of, I guess, synergy in this choice. And so specifically, they had a particular, they make these curli fibers. They can also make them recombinantly, so they can engineer that curli protein to bind other things. And so in this example, they were able to make them glow because the curli protein had like a tag, not like a tag, had a tag on it that allowed it to fuse to something else, to another protein that they put through. And then that gave it this glowing property. But you can imagine they have what they're showing off here in a way is their control over the system, that they can take curly eye fibers and make them do different things. That's very promising from the engineering standpoint. It's all about control here. I feel like they're not so much discovering the mysteries of nature as they are like making the mysteries of nature do stuff for us so that we can make objects.
That's right. And so the idea there was, I think this, so there to highlight the point that we're trying to go for a pseudoplastic with this.
Yeah.
And so
I was gonna say, and they show it because they can take that gel that they made and they can put it in a machine that measures these things, and they get these graphs out of it. I think we'll talk a little bit more about those readings afterwards.
Yeah, I think we'll, and essentially what they kind of find is that cells can affect like the matrix so they, in this case, they use mucin binding domains, and these matrices were thought to have like shear thinning properties.
Yeah, so this paper, I mean, this is just interesting sometimes to see like the progression of where people's work goes. Because in this paper in 2019, the reason why they used mucin binding domains was because they wanted to have they weren't even printing at this point, right? They were just making sticky E. coli that they could like control under different circumstances. They wanted this E. coli to stick inside mouse guts. And so like, okay, look, they can make E. coli stick in mouse guts. And what they were able to do was then take these rheological measurements and show that they had some interesting properties in terms of shear thinning. Yeah.
Yeah, so now we're finally getting to this paper where we've got figure one part A, where we're looking at what they've kind of engineered. So they're engineering these curly fibers are nanofibers, and the idea is they've edited them somewhat. So they've added so these curly fibers are in red, and they've added some domains called CSGA alpha and CSGA gamma, and these come from clotting proteins, right?
Yeah, from fibrin. So and they really just did a biomimicry design style here. They were like, oh, we know about a type of fiber that creates itself spontaneously inside of a system. It's a blood clot. And so what makes that so structural is the question they ask themselves, or not the question they ask themselves, that's the inspiration that they wanted to draw from. And it's actually quite a simple system. It's just these two cross links, the CSGA and the CSG gamma, or sorry, what did they call it?
They call it the knob and the holder domain from the fibrin.
Very, very theoretic, very understandable. Knob and hole. The knob is alpha, I think, and the hole is gamma. Or no, the hole is gamma, and the knob is that knob is alpha, and the hole is gamma.
Yeah. So in this, like the blue dots and the kind of green, blue dot is the knob, and the green kind of C, backward C is a hole.
Yeah, and we saw before that they were able to make fusion proteins. So CSGA is that curly eye monomer, and so they're going to fuse. They're going to make two monomers, one that has the knob and one that has the hole. And the theory behind that would be that then those two curly eye nanofibers would get intertwined in a specific way, and that might make a stronger link. And I think they were inspired by that mucin domain stuff, by the fact that live cells in the mucin thing seem to be increasing its properties. Yeah, this is really just, who knows how many things they tried that didn't work, but certainly after it's like extracted from their success, like you can see how it was a type of inspiration that made them go down this path.
Yeah, and so this kind of shows that their design ideas behind this, and what explains what they're hoping to do with the rest of the paper. So that brings us to table S1, which kind of goes how they're designing their proteins.
Yeah, so this is the fusion. These are the fusions that we're seeing, in more detail. And then importantly, it also, they need to secrete this. So this is not intracellular, and this is not lysing the E. coli cells to get this protein out. E. coli knows how to secrete curly or curly, and it's able, and so they make sure that the signals are in place so that it can do so.
Yeah. So we get like the first test. So this is where they look at the fibers under electron microscopy.
Mm-hmm.
Oh, yeah, I think must be given.
Yeah, it's electron
I've got
microscopy. It's using negative staining, so they just have something that fills in the gaps, right? And like the ideas of negative stains is that the stain doesn't bind to anything in particular. In some ways, it's where the stain doesn't bind is where you see the little lines. And
Right.
so, yeah, they have a bunch of this gel, and they look at it, and they can measure the width of these fibers. And I guess interestingly, in the CSG, when you mix the two fibers together, or I guess it's probably E. coli that's expressing both of the constructs. When they have the constructs that express them both together, they see like a thicker fiber. And so their thought is that, oh, it must be that, the single fibers are thin, and then what they did is working because it makes one thicker fiber.
Yeah. So that's, the thick fiber looks better. So you need to have both knobs and holes in order to have these thicker and presumably like kind of more, maybe stronger. We don't know yet. That's going to be something that we have to look into. Right. And that's what these researchers are going to be testing. But they do notice that there are there is a mixture of fiber diameters. So there is some diversity there. So some of the big ones are 10, but I think that's like the upper limit, but there are small ones.
Yeah. Well, maybe it also shows that it might not be 100%. Like, just because you can get thicker fibers doesn't mean that there aren't the thinner ones also in that same mixture. And I mean, that again is maybe, or that's something that I feel this is very engineering. It's like, right, they want to know like, oh, how pure is the material that they're working with, because like that's going to affect its properties and so forth. So this just gives you an idea that there's some heterogeneity.
Yeah, and so what they did now, they can filter out the cells and just look at the hydrogels themselves. So again, we've got your CSG alpha, that's the knob, and gamma, which is the hole, and then alpha gamma, which is both the knobs and the holes.
Yeah. And this time they're looking at it with SEM are these images. So they aren't taking slices of the ink material or the ink cultures. This time they're sputtering it. They're coating it with some metal, and they're looking at what that looks like. That's why these images look a bit different.
Yeah, so these images give you the texture of the fibers, whereas the previous ones showed you kind of their distribution. This one looks at kind of how they look like when they're bunched up together.
And I think it's really cool, too, that they give us the optical images as well. It gives you a sense of what this material is, right? They put it in the bottom of a bottle, and they flip the bottle upside down like a blizzard, if you guys know Dairy Queen. So just...
We don't have that in the UK at the moment.
Well, it's like some viscous ice cream smoothie, right? It doesn't fall out of the container because they can flip it on upside down. Wow. And so that's the demo. They're using the same demo that's used in that corporate ice cream chain is being used here in the scientific paper to show us how sticky this material is.
I mean, horrifying, but I can't wait to try it. But yeah, no, you're right. Even the simple act of turning these things upside down demonstrates that these materials are that level of viscous where they won't immediately flow downwards. So yeah, how do we measure this viscosity? So I'm using this example of the double stuffed Oreos because they've got this machine called a rheometer, which is this kind of spinning disc which kind of you put your material in between, and it measures the kind of the feedback, the force that it puts in. This particular image is from a completely unrelated experiment where they tried to look at the viscosity of worms. The idea that worms produce mucus, but we're not talking about that right now. It is gross.
I'll say what's nice about this sort of machine in order to measure viscosity, like when we looked earlier at the example of something flowing through a pipe, you can see there's like heterogeneity in that. Like if we imagine all the different layers, the middle is going to have faster flow than on the outsides. But in this system, right, the whole liquid is experiencing the same force, right, that's going through. And so they use oscillatory motion. Yeah.
Yeah, it's interesting because the bottom disk isn't moving, the top one is. So, in fact, you've got two boundary layers where the top is kind of like pulling the first one along,
Mm-hmm.
and the bottom isn't being pulled along. So you can see the ones at the top are kind of being flung through there much faster than the ones at the bottom.
Yeah.
So the rate of difference between that is kind of a measure of how viscous liquid is.
Mm-hmm. Yeah, and what comes out of these machines is two measurements, the G prime and G double prime. This is a lot. Like G prime is a lot like if people studied springs in like high school physics, right? It's the way in which a spring has force that returns it back into its shape, and that's the elastic component. And then G double prime is is the one that is more about the liquid part of it. That's like whether the how I guess it's almost the viscosity style, right? Like how much it like flows. And so like you can take the ratio of both to get some sort of understanding. They don't do that here. They just give us like they just report both to us. So there's two lines for every sample here. One gives us the G prime, one gives us the G double prime.
Right. So I'm not sure whether I've interpreted this correctly, but I feel like if you're lower, then that's more watery, and if you're higher, that's more viscous and more kind of elastic. I don't know whether I've got that right.
I think that's true for one of the lines. It's not true for both of the lines.
Right.
Yeah, it's not true for both the lines. But
okay.
I think at
Sorry, physicists.
Yeah. Because like they don't show the ratio on this particular graph.
Right. That makes sense. So we've got we've got like a couple of things where they measure the rheology of things. So they
look at frequency. So they can look at the response. I don't know how the machine works exactly, like in terms of how does it actually give us these numbers. It does, as you said, have something to do with how much force they're applying on the spinning object. But there's also frequency, right? They can spin that thing faster or slower. And again, like it's attached to like a motor and a sensor system that knows how much force it's actually adding into that particular into that system. But you can see in B, I think B is where you see it has some of these potentially shear thinning properties, because as they add more strain, right, as things get displaced more, the moduluses are changing, right? So it means that the material doesn't have a consistent property, right, throughout all of the different ways it can behave. There's some point at which, like, I guess it's a break point in some ways. You put so much strain on this thing, it's going to start acting differently afterwards. And that's in line with the shear thinning idea, but I think in a later figure they have like a more definite shear thinning example.
Yeah.
This one is like a more definite shear thinning example.
Right. As I understood this, like viscosity is the idea of things. So higher level, more viscous, right? So, and lower level, less viscous.
Yep.
So as you increase the shear rate, it becomes less viscous.
Which is great. That's exactly what they want. And so I guess all of that G prime G stuff, like it's all coming. They're not doing separate experiments. They're not doing separate experiments here. It's all in that machine. But I guess you can think of them as like they're like the components of viscosity, right? They're like component measurements that, like, when you sum them up in a certain way, they come to viscosity. But yeah, this is the figure that in the paper they say, oh, this is really, we're seeing shear thinning. But when you look at the way that they did it before in the live cell mixture, in the mucin binding one, they were already looking at shear thinning just by looking at those G prime and G double prime curves. So you can sort of figure it out from both.
Right. And so like, they kind of put these into graphs where you've got like the yield stress and the shear modulus.
Well, this is more now to do with, because like these are now at single, like they have to these are like fixed measurements now. It's not over like a range. So these are, maybe you can think of this as like the material properties itself, like the stiffness we're getting more at stiffness now, a little bit here.
Right. And so basically, like the knob and hole motifs, when they're put together, they produce a stiffer material, which is kind of what we'd hope for this.
And this is like because when they first did this experiment, they were co-expressing the CSGA fusions both in the same bacteria, and then purifying out the resulting hydrogel mix. But you know, you could just ask like, but what happens if you just mix them 50-50, right? Like you made some CSGA mix and you made some CSGA alpha and gamma mix, or separately, then you mix them together. Are they the same thing? And it seems like the same thing. So there isn't anything, they weren't introducing, I don't know, some unknown variable, right? By like expressing it in the same.
Yeah, I think that's an important like kind of consideration for if you're looking at future and looking at kind of proteins that are going to be more like kind of avidly binding to each other. So if you end up with something so stiff that it immediately is hard to get out of the nozzle, then mixing two bacteria together that can do it might be.
Oh yeah, I guess yeah.
I don't know.
But actually, I would say that that's not true, because if you go back, if you go back a figure, so like in this figure C, they have CSGA and CSGA gamma by itself. Like that property is actually the same, right? The shear thinning property of those two are the same as the shear thinning property of the one. Or maybe it's like slightly different. I don't know. Maybe it's significant if you zoomed in on the lines, but like to me, those lines are kind of the same. So it's not like the shear thinning that changes when you add it. It's just the end stiffness of it all that's different.
Okay. That makes sense.
So again, like going back to that concept, like that I kind of want to say, like it's about like these are about like the components, like all this talk about like these different moduluses. They're all like properties of these materials in a really specific manner. But at the end of the day, I mean, you lifted our little banner.
I did lift
right?
up.
Like,
so announcing that now it's time for 3D printing.
Yeah, at the end of the day, like what we're gonna see, like our results is like in an actual test of what we want to use these objects for. So yeah, that's what they do.
So yeah, they're tested on a modified 3D printer, able to extrude the gel. So these are kind of images of the gels that were produced based off that, and at different pressures as well.
Yeah. Yeah, they kind of they just want to try a whole bunch of different things. To me, I guess I like to just make people. I'm sure the physicists are also pointing at biologists and being like, biologists like to do this, engineering, right? Like everyone has their own idea of how people approach their discipline. To me, I'm like, oh, this is such an engineering thing. It's like they just want to see like all the different ranges in which something occurs, and then choose the best one, the one that works for them in order to achieve their goals. And I think what's interesting about this paper, though, is that there is no pre-specified goal except like making a good 3D print. So actually, all of this data that they gather could just be useful, right? When a goal is set for the particular printing modality, you know the range of things you can try in order to get what you want.
Yeah, and I think so they try different flow rates and peak pressures, and I think mostly they find that the diameter of the knob and hole motif tends to be a lot thinner than the ones that don't bind together.
Yes, yeah. So that would indicate, I think that's a direct, that could be predicted from how stiff the material is, because it means it's not spreading out. In some of these, the more you, like, in the other two examples, they spread out after they've sat down on that surface. But because the knob and hole together is stiffer, it's just thinner. And that is a desirable property, right? Because they want to be able to print out the shape and have that shape stay and not, like, I guess, get mushy.
Yeah, and I think they tested this across a bunch of different pressures.
They did it all.
V rate. Yeah.
So, I mean, I guess I don't know,
And
like in terms of design, maybe one of those is good, right? Like maybe they would want one that spreads, but like, they get to make the choices, right? The idea is here, they're gathering all the data to make choices.
yeah, I think the idea is that if it can hold better, if this string can hold up at a thinner diameter, then it's more structurally stable. Like kind of, whereas a thicker one, you can't get it to hold at small widths.
Mm-hmm.
I
Mm-hmm.
think.
Mm-hmm.
But yeah, I think we've got like a video, like movie figure S1 is like coming up next, which will show a lot more and about how they kind of test the structural stability of the strains.
Mm-hmm.
Um, Hopefully the video will start to play there.
Yeah, this is their cut,
Yeah,
taken from the supplemental figures.
yeah. So they test the structural stability using this kind of drawbridge test, where the idea is that if something is strong, you can like leave it out, like create a drawbridge out of it, and it won't snap.
Mm-hmm. Mm-hmm. Yeah, I mean, it's like this is a classic physics but engineering thing that's done in grade school, right? You have to build a bridge out of toothpicks or whatever, and then
Yeah,
you learn about like how triangles are really strong. Well, here they just like they're just drawing a line of their material, and that gives them the same output. Um, this is now they're like then they also use it to print shapes, specific shapes,
yes.
just to see how well the shapes look. Or I think one of the things it's proof of principle, right? Like
Hmm.
if they can make these shapes, it means that their ink must be meeting some minimal criteria, and they feel confident because they've done the rheology. They know that it has particular properties.
Yeah, particular set of skills. And yeah, it's really great that we've got get these videos now of these prints actually coming in, and they they get the idea of like different props. So you got your three-dimensional prints that you had previously, and now they've got getting a two-dimensional print to kind of like demonstrate that they can maybe create a plaster of some sort or like some kind of
Hmm.
functional living material.
Yeah, like this is like a band-aid, right? Like, oh, they can
Yeah,
make a band-aid out of it. I also found it weird though in these videos, like that they're holding it with their hands, like
yeah,
that they didn't like tape down. I don't know why that is. That's probably. I bet it has to do with like maybe not having a camera rig or something. And yeah.
yeah. So, like again, so we're going back to what they kind of showed in the video where filament collapse. The idea that you can create if the material's strong, you can create a drawbridge out of it. But let's say if your material's not strong, then the drawbridge collapses, and we kind of see that with the like knob and hole domains on their own, where you get these bridges just kind of go and
Yeah. Well, I think they can even predict that they're able to predict a property of the material just by looking at the angle of sag on different
yeah,
gaps. Because as you can see in their little force diagram, right, like all of those forces can be accounted for, and then that gives you an idea of the strength of that. And so, of course, very, I guess, dramatically in the CSGA, in the monomer, in the knob and hole monomers independently, those collapse. They can't span the bridge, right?
hmm,
They don't have the stiffness or the sort of internal strength to withstand the force of gravity pulling down on them across those distances. So that's like a nice, like, it's just a dramatic way of looking at it. But, they also can use some math and make a graph out of all of this.
yeah. And that's what they do. You've got a little
Uh-huh.
graph of like of how saggy these knobs and holes are
Hmm.
on their own.
And they made a comment that like it's not 100%. It doesn't match their predictions because like you can measure from the SAG, but then you could have also plugged in the rheology stuff that they knew ahead of time, the
Hmm.
yield strength and things, and then try to predict this. So like they do notice that there's a difference. And I guess maybe that's just a nice lesson. I think they explained it away in some reason. I don't remember what the explanation was, but I want to bring it up because like this is like a good thought process behind engineering in general. Is that it's great that you can make predictions using the properties that you've grabbed, but at the end of the day, a good experiment like is gonna give you exactly what you need to know. So you want to make sure like even this is very abstract. Like there's so much abstraction here in terms of making this material at the end of the day, what we want to know is like for the specific purpose of making something, is it going to work? But in this paper, they're sort of building us up to just give us confidence that, yeah, we should use this thing because like it is pretty good.
Yeah, and they've constructed various shapes from several layers of this ink. So we kind of saw like two of these from the video, the hollow square and the hollow kind of circle. But we've also got this crisscross pattern, and my favorite is this pyramid, this weird like cone thing they created.
Yep, and it's just a demo. Like, oh, look at these cool things that you can make. Like when you throw up your 3D printer at home and you go to Thingiverse and you pick up your favorite things just to make sure that it works. That's essentially what they did here.
Yeah, that's kind of a lot of the fun of this, where like now you've got this like product, you can think about the applications. And so, for instance, they're thinking about therapeutic level material, something that can house cells that produce drugs. So I think in this case they got like a strain of E. coli that produces a drug called azurin. I think that might be an anti-cancer drug.
Yeah, they said it's some sort of anti-cancer drug. So I guess this would be, I'm trying to think of like what the application is. Anti-cancer drug inside of a patch. So like maybe you just want to slap on right this like particular thing. Maybe they're imagining it as like some sort of like nano, like printed at a really small scale and like flowing through your body in some ways. I don't really understand. I think it's like, this is also very abstract to me. I think it's like they can make something that they can make a bacteria that makes a particular compound and put that inside of this material. Whether or not you would ever need that, like, would you ever need this particular drug in this particular ink? I don't know if that's the case. It's more like they can make things inside of the ink. Yeah.
Yeah, I mean, part of it is like what you can have available in your lab and what's detectable. In this case, I imagine that they had this available, and they figured out how to make it detectable. So you got this Western blot where the black spots are essentially the drug's detectable, and the ones where you can't see anything, drug isn't detectable. And they use like IPGTG, which is a chemical that signals and tells the cells, "Oh, please make this drug." And so you can see kind of how leaky that is.
So I think,
and so this kind of
I was going to say it's nice. A nice thing that you learn from this is that the gels are infiltratable by IPTG, which I guess might be useful to know in terms of if you do want to turn something on or off, like you want to know that your signaling molecule can get into those cells. So that's nice.
Then we've got like the idea that you can maybe sequester toxin in materials. So I think we did talk about this a while ago with the kombucha sewage kind of filtration, where you can kind of use like bacteria and bioremediation, where you can train bacteria to take a material that's toxic and digest it and get rid of it. So in this case, they've got like this compound called bisphenol A or BPA. That I think they've trained bacteria to have this gene that can sequester BPA, and they use it. It also when they demonstrate these, they're doing different patterns as well, which I'm quite interesting.
Again, like I don't really understand what the interplay is between the pattern and like the thing that they did. To me, it just seems like they're doing it to show you these variabilities, but whether or not this is the actual application they're imagining is more abstract, right? Like the actual application, as I said, we would have to see like a real proposed use. Like what is this shape doing that they've created, right? That does a certain thing. You mentioned that that kombucha paper from a while back. I think in that kombucha paper, they were making filters. And so it was more that the filter had a good property to resist getting clogged up. And they thought that was because it was made of bacteria, that there were like living things in there that like wanted their own lives. So like they didn't want other things to live on that surface. And that was a nice like feature of that material. So actually I would say that was more similar to the last one, right? The kombucha was maybe more similar to the last thing, right? Where you're like, oh, like you could make a filter out of this ink, and that filter could make an antibiotic that prevented stuff from growing on it. Right, like that would be like the technology similarity here. Of course, in the kombucha thing, that is in that weird cube of engineering. That kombucha thing fits. It's not 3D printing though, right? That's the bacteria made it itself, right? The bacteria made the structure. Here we control that structure. So maybe it's useful if you had like a very specific shape of filter that you needed to make, right? And like you couldn't get bacteria to make it in that shape, so you could print it in that shape, but you could still have bacteria living in it doing some function. Yeah, but then the second example, this is like a remediation example of some sort, where now whatever you printed, it's absorbing stuff from the environment. I don't know if I don't know if we've talked about like this type of technique of sequestering things, but
yeah, I don't think so. I think this is like the first time we've talked about that, but I think it's something that comes up occasionally when taking out chemicals. I know there's like some stuff on like excreting heavy metals in the soil, like how like they train some plants to like mine for gold essentially by sucking up gold particles through their roots.
Totally. I mean, you could say the same thing about this, right? Like you could imagine, again, this is all very conceptual, this paper, right? Like it shows us the beginnings of the technology, and then it wants us to take our imaginations and go like, oh wow, look at all this cool stuff. But then you have to realize that like that cool stuff needs their own sets of papers to understand what's going on. But yeah, like you could imagine the same thing here. If, instead of making something that sequestered BPA, what if you sequestered something else, right? What if you sequestered metals? What if you sequestered something you wanted to harvest, right? So like this doesn't necessarily mean you want to take things. This demonstration isn't necessarily saying you can take things out of an environment because you don't want them. You could also take things out of the environment because you do want them, and that could be really useful, right? You can imagine all these different shapes might fit inside of different bioreactors in a certain way. It might make something reusable because it's in a certain shape, right? You can clean it off and then take out like extract all that BPA or whatever you wanted out of this and then put it back in the environment, right? Use it again to take things out of the environment.
And I guess that brings
respirators.
us to our final example.
Yeah, no, I like this. I like this thought in the chat. Yeah, like right, like this could be respirator medium, right? Like those are things where we want to filter things out. So like, what would it be, right? Like, what sort of particles would you want? COVID.
Yeah, so things that'll filter out COVID. So you got your biofilm that has like ACE2 proteins all over it, and then
yeah. I mean, that was like the chewing gum, actually. Now that this has come up, like a while back, we read like a news story that said that they put ACE2 in a chewing gum, and so that you would chew that gum and somehow deplete the amount of SARS-CoV-2 in your saliva.
Yeah, debulking COVID with like a chewing gum made from chloroplasts that were designed to secrete ACE2. It was a very fun paper. We look, it's in our back catalog. I'm sure we've got it in. I put it in the title, definitely.
But again, like that is the same. Like I think that's the right way of thinking about this particular application. Is they're saying like what draws those things out. But again, like I kind of like the harvesting angle a little bit on this one, because I can imagine like harvesting, like printing out the structure to harvest from like just like supernatant, right? Like what if something was secreted in supernatant and you wanted to like adhere it to a structure? Like you could print sponges of this material, right, and dip it into that supernatant and then take it out, like just like how. Oh yeah.
And then I guess you'd like pop those bacteria to create more supernatant. Then you have to extract it out again.
Yeah.
But
reusability. I mean, this is an interesting question about reusability. Like, I don't know whether or not like the gel is more reusable than another modality. And I think that this is like where the rubber meets the road, right, is going to be doing like comparisons. Is this technology better than something that already exists that is doing things great? That's like a complicated question because it's going to matter like what specific applications we're talking about.
Yeah, I mean, I'm gonna go there. 3D printing ink, very artistic.
It's a skull and crossbones because in this particular example, they are actually trying to kill the bacteria that are inside of the matrix. And by the matrix, I mean like the e-ink matrix, not like some other matrix. So this is cool, right? Because they want to show that they can, that they can kill off the cells that are in this particular thing, because maybe you don't want living cells all the time inside of your e-ink, but you want them sometimes, and so that's what they're showing here.
Yeah, and I think that's the final like figure that we're showing. Yeah, and I do want to circle back to those, but yeah, there are certain questions about like kind of like how long do these, how stable?
I mean, they're stable in the sense that they can make the shapes that they want. Are they stable in that those shapes can then go into their target environment to do the action and still come out intact?
Yeah. And I've got like other questions about like, say, let's say you print out your wonderful 3D shape and then let the bacteria grow more. Will that, won't they eventually change the shape of the whatever you printed out?
Well, the bacteria, yeah, I mean, that's an interesting question, but they show that they can control the growth of the bacteria in there. So like that is like, I think they're definitely speaking to that skepticism of yours, being like, oh, but don't worry, like we have control of anything that grows in that in the shape.
Yeah, they control the horizontal and the vertical.
Yeah, welcome to the outer limits of e-ink printing.
Yeah. Yeah. I think because I think the idea of like having like this is essentially like 3D always renewable, because you've got your bacterial structure and then you can kill your bacteria off at the end of it.
Yeah, and maybe that could be something from the safer perspective that is nice about e-ink, is that so when we make these objects, like, oh, okay, it's great if you want a jelly, you know that it's going to degrade afterwards. You don't have to worry about the sort of the circular, right? Like, what's the life cycle of plastic after you've printed it out in these different ways? Yeah, they don't really do a lot on that front. I wish they did actually. Like maybe as it like still maintains its shape, because I think that all of these are wet.
Yeah.
Nothing here is dry. Everything's kind of moist. This is all like a jelly thing. And so, yeah, I think that's another really interesting perspective or like direction that the technology could go.
Yeah, I mean, I think that there is a nice idea of something having a renewable substance, and I think I mean, it's been quite interesting, like learning so much about like because a lot of this paper is about the properties of the 3D gel and like kind of why they need the specific viscosity and how they measured to see whether they've got it.
Yeah. Yeah, I wanted to just bring up that point because like I was like, oh, cool, yeah, this could be like a filtration medium. But like when people do filtration medium, the size of those features is really small, right? Like the size of features inside of activated carbon or whatever, those are like our standards of filtration medium. Those are like at the nanoscale. Like these are nanofibers, but they're printed at the millimeter scale.
Yeah.
So so like we have to think about things that are at that scale.
Yeah, because we're not the ones printing out the fibers. It's a bacteria that are printing them out. We're just printing out the bacteria and hoping that they print out the fibers in the way we'd like them to. But there is a level of control that isn't necessarily possible at this stage.
Well, like it's not like, I guess what I'm saying is it's not the 3D printing that is giving us the nanoscale things. Yeah, the nanoscale, and maybe that's a little bit, their title is misleading in that way, or at least it's bringing us the hype of the nanoscale a little bit more, because like the actual print is not that. I'm pretty sure, right? Like if we go back to where they printed the fibers under all the different pressures, that was a millimeter scale, I'm pretty sure.
Yeah, let me pull that. Well, yeah, that was I think that was a millimeter scale. Let me pull that up quickly.
Yeah, of course. Like how do you get a nanofiber out of a 3D printer? That's that doesn't. 3D printers do not have that that power.
Yeah, indeed. So yeah, these are like the millimeter scale. So they. Oh, okay. 1 minute. Sorry. Technical issues.
Yeah.
So yeah.
Yeah, these guys.
Yeah, so we get a nice impression of like how thin they get, but yeah, this is very much on the like the millimeter level.
Yeah.
Um,
So, I mean, there could be interesting ideas for like thinking more about that like sequestered toxin. That it does sound like that's like a filtration style thing, but like, what do we really filter at the millimeter scale? Um, that's like the mesh grates before, right? Like you can make like a sieve,
yeah,
a sieve that was activated in a certain way. Um, maybe
yeah, I
like
think the
oh, go ahead.
sorry, because I'm thinking like the more important, like the kind of the production of bacteria that do different like kind of metabolic things,
Oh
and
yeah,
then
and then you flow something through the bioreactor, and as it's going through one set of sieves, right, it's getting transformed in some way. Then it goes to the second set, and it's getting another transformation.
yeah,
Yeah,
you produce like a thing that like that's transformed by one bacteria and the others, and then you can kind of almost have if you've got like a temperature gradient and other things, you can kind of yeah, I mean you
yeah,
can handle like biochemical reactions on well handle bio in a different way. I mean, if you're
yeah,
thinking about
yeah,
maybe
absolutely.
yeah, oh
Yeah,
well,
and again, like in all of these interesting applications that we're thinking of right now, or that you might be thinking of at home, at the end of the day, like this paper like is attempting to spawn those in us, I guess. Like that's it was very much written to be like, oh, think of all the things, but then you actually have to do those things. And you have to try them, and like they might not work for different reasons, and there might be better technologies out there to do the things that you imagined, because like that's how the world is, right? Like there's lots of different ways to accomplish the same goal. Like what you were just saying there, Faz, it reminds me that we've talked about synthetic biology techniques about separating different metabolisms or binding different metabolisms together, right, in order to have groups of bacteria do different things. This is like, in some ways, like another tool in that spatial separation toolbox. But whether or not it's going to give us like a really great pathway to make something cool, that remains to be seen, right? Like in its own separate proof of concept.
yeah, and I think this is very much like kind of an intermediary paper. We're not it's not quite there yet, but they're toning up the technologies to make things better.
Totally, but it was a cool read. I didn't expect to like reading it actually that much, because I saw that it was a very engineering forward paper. But actually, it's nice to learn a little bit of rheology and see like what people are trying out there. And it also actually feels very doable. Like I almost feel like I want to try to make some of this sticky stuff at some point, because it is just E. coli expressing this one construct,
Yeah,
and it secretes it fine. It's in the medium, and then they just do a bit of biochem, and they get this sticky material.
yeah, that'd be a great little project to have. Um,
Okay, well, join us next week for more cool microbiology news.
yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible, nay, likely that we didn't get everything right, especially the physics stuff. My bad. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process that we can all participate in, so we hope you've had a good time listening us ramble about microbiology and physics today. If you found something unclear or you have a question, just let us know in the comments.
Yeah, it's been a pleasure chatting with you, Danny. And thanks to all of our lovely commenters who have been very supportive in the chat.
Totally. See you next time.
Yeah, tune in next week for more microbiology content. Goodbye.